In today's lesson, we are going to look at the next stage of going from a gene to a protein, where we go from the mRNA to the polypeptide. So we start with the messenger RNA, which was produced in the nucleus and exits the nucleus through the nuclear pore. And it is read by codon. And remember, this is those 3 letters, and that would be RNA letters that are going to specify for different amino acids to be added to a growing polypeptide. This is done at the ribosome, and this is the assembling one amino acid at a time as one codon is read at a time. Who brings these amino acids? tRNA brings the amino acids to the ribosomes, and this is how a new polypeptide is made. So let's look at the tRNA. The tRNA is RNA, so it is single-stranded, but there is a lot of complementary bonding within that single-stranded RNA. So we end up with 4 double helical segments: 1, 2, 3, and 4. The tip of the tRNA contains the anticodon. So this is complementary to the codon of the mRNA. So, for example, if this is the anticodon UCA, the codon that this is going to bind to on the mRNA would be AGU. Lastly, we have the amino acid that is attached to the top of the tRNA. And this is going to correspond to whatever codon it is coding for on the mRNA. So whatever mRNA that this is going to complement, this is the amino acid being coded for on the mRNA. Now, the cell is very smart in the sense that if you look at how many potential codons, there are 61, which would mean we would need 61 tRNA. But luckily, there is this wobble effect. And this shows, or this is done by the pairing of an anticodon with only the first 2 nucleotides being very specific. So looking here, our anticodon is UCA. So the first 2 are going to very specifically bind to the codon with a G. But then the third one can be variable. So it's not necessarily going to be a U, it could be something else. So let's look at serine. This is one of the tRNA for serine. So looking here, we see serine could have these codons. So looking, we have the UC at the beginning, UC. But then the third one could be either UCA or G. So the tRNA for serine could have the anticodon AG and then something else. Now similarly, there's 2 more codons specifically for this tRNA that we're looking for. So this tRNA has the anticodon UCA. So we see this right here, the top one, AGU, is very specific to that anticodon. So this codon is exact complement to this anticodon. But if we look just below it, we also have AGC. So this too will bind this tRNA. So looking at that, how many tRNAs would serine have? 2. So 1 tRNA is going to bind any of these 4 sequences because the first 2 are the same. And then another tRNA that's going to have to be specific to the first 2 of this codon, and then the third one, it doesn't matter so much. So you're going to have potentially 2 tRNA for this serine. So let's look at our codon chart. This is out of your textbook, and I just want you guys to slowly get familiar with the names of the amino acids, their 3-letter abbreviations, and preferably even their 1-letter symbol that represents them. So let's go ahead and look at serine. So first of all, here is serine here. So this is the codon chart showing the codons for serine. So we have the U, C, and then any of the 4 third nucleotides are going to call over a serine tRNA. We also have down here this tRNA. These codons that would call over a different tRNA, but still bringing over serine, and that's AGU and AGC. What I want you to think about is the similarity between a uracil and a cytosine. What are they both? They both belong to what category of nucleotides? Pyrimidines. They are both pyrimidines. So they're the smaller, they have the bigger name. They're both pyrimidines. Let's look at some other examples over here. So we have tyrosine here, and look, it has the same beginning, U and A, but the third has a U and a C. So those are also both pyrimidines. Histidine, first 2, the second one are both pyrimidines. If we look down here to glutamine, first 2 are both Ca, but then the third one is a purine. So these are the bigger ones. Ag, that's big. Okay, so if you look through there, it is, again, UC, we have AG, UC, AG. So that helps if you're wondering how asparagine isn't being brought in instead of glutamic acid. That's a lot of it has to do with that third nucleotide being a purine versus a pyrimidine. So that helps with that. So as another example, let's briefly look at tyrosine. So here's the tyrosine tRNA, and the anticodon is AUA, and there's only one tRNA for tyrosine. And it can bind to one of 2 codons, UAC or UAU. So UAU is a perfect match for the AUA. They're all complementary to each other. So this one is like a perfect match. However, this tRNA carrying tyrosine will also recognize and bind to the UAC codon. So the C isn't a perfect match. A wants the opposite of A would be AU, but being in the third position and the fact that A is also a pyrimidine, just like the preferable G would be, this tRNA will still bind to the codon bringing tyrosine. So this flexibility, again, is going to give the cell the ability to have less tRNAs. So let's talk about how we are producing these tRNAs carrying amino acids. So the addition of an amino acid to a tRNA is called aminoacylation. Aminoacylation. It's also known as charging a tRNA because you are kind of giving it its energy that is going to be used to grow the polypeptide. So aminoacyl tRNA is the tRNA that's carrying its amino acid. So previously I've just been, and a lot of times it will just be called a tRNA, but to be specific, it's a tRNA without the amino acid. So that's the only time you would actually properly call it a tRNA. Once it's carrying the amino acid, then it's actually called an aminoacyl tRNA. So there's actually 20 different aminoacyl tRNA synthetase enzymes. Aminoacyl tRNA synthetase enzymes. There's 20 of them, one for each of the 20 amino acids. The energy in the aminoacyl formation is used for the production of the polypeptide growth. So the energy that is going to be in this amino-acylated tRNA will have the energy that's needed to add the amino acid to the growing polypeptide. So in this diagram, this is our specific amino acid tRNA synthetase, and it has a specific binding spot for that anticodon. So that's how it knows specifically which tRNA it's supposed to be binding. And so we have an amino acid here, we have a tRNA, they're going to bind to this enzyme, and then we have our amino acyl tRNA. Okay, we talked about ribosomes in our introductory lesson. So they're made of two subunits, the small and large subunits. This is the small subunit. This is our large subunit. Each subunit is made of protein and RNA. It contains specific binding sites that bring together mRNA with amino acyl tRNAs. And we talked about those a little bit in the introductory lesson, and we're going to talk about each site in our next slide. So the mRNA threads through the ribosomes. Remember, what is it that is going to be recognized by the ribosomes? What has been added to an mRNA to make it recognized by ribosomes? Should be able to answer the 5' cap. So here, there'd be a 5' cap. So this is our mRNA in green. We have our small subunit and our large subunit. This is a polypeptide already in action. So we have a tRNA that is exiting. We have a growing polypeptide chain. And then we have an incoming amino acyl tRNA carrying specifically serine. So first we have our A site. This is the amino acyl site. This is where we have our incoming amino acyl tRNA, and this is where it is going to bind to the mRNA here. So we have our anticodon, and we have our codon that is coding for serine. Our next site is the P site, the peptidal site. So this is where we have our growing polypeptide. And it is bound still to this tRNA, and the growing polypeptide gets moved and added to this tRNA. And then lastly, we have our exit site, and this is where our empty tRNA is released from the ribosome. And it will go back and undergo another reaction to pick up its amino acid. The first stage of translation is initiation. So this is overall initiation is where we have the combination of the mRNA with the small ribosomal subunit and the addition of the first tRNA containing methionine. So let's talk about how that happens. So we have our amino acyl tRNA carrying methionine forms a complex with the small ribosomal subunit. So this is how it starts. We have these two together, and we know methionine is going to be the first amino acid because that is the start codon. So they're bound and ready to go. Next, this complex binds an mRNA, and it was recognizing that 5' guanine cap. Remember, there's how many? 7, yep. And then it's going to move along the mRNA until it meets that AUG codon, which is the start of the polypeptide. So this is called scanning. It'll literally scan the mRNA until it gets to that AUG. Then we have our large ribosomal subunit join in. By the end of initiation, we have our methionine tRNA in the P site, because this is the start of the polypeptide. The reading frame is the series of codons for the polypeptide that is encoded by the mRNA. So it's essentially not including the G 5' cap or that poly A tail. The next stage is elongation. And so this is what the animation in our introductory lesson was exemplifying. So we have our methionine tRNA at the P site. We also have a second mRNA that's coming in and is going to bind at the A site. GTP, which is like ATP, is hydrolyzed to provide free energy. So we're getting free energy from GTP. The methionine is cleaved from tRNA in the P site, and then it forms a peptide bond with that second amino acid on the tRNA in the A site. So it's moving from here, and it's going to get added to this amino acid. This is catalyzed by an enzyme that's part of the ribosome, and that's called peptidyl transferase. Peptidyl transferase, because it's going to be transferring whatever growing peptide there is to the new amino acid. So peptidyl transferase. The ribosome moves in a 3' direction to the next codon. So it's going to be moving towards the 3' end of the mRNA. The two tRNA remain where they are, so they're not going to move with the ribosome. They stay attached to the mRNA as the ribosome moves, as we saw in the animation. So now, the tRNA that was in the P site is now in the yeah, that happened, is now in the A site. And, what was in the P site will now be in the E site, and what was in the A site will then be in the P site. So we have the empty tRNA in the E site, and that will then leave. We have the growing polypeptide now in the P site, and this A site is ready for the next amino acid to come in. Lastly, we have termination. So the A site arrives at a stop codon eventually. So UAA, UAG, UGA. So a protein release factor binds there instead of a tRNA. The polypeptide chain is then released. The ribosomal subunits separate. The empty tRNA is released, and the release factors are also released. So it's just an immediate just dissociation of everything. And then everything will go on and start something new. So let's just talk about some similarities and differences between prokaryotic and eukaryotic cells. So in both, multiple ribosomes can translate from a single mRNA at once, increasing production. So you imagine if this is our mRNA and we have a ribosome bind, as it's moving along, another ribosome can come along and bind and start moving along. And then you can also get like a third one come along, and all three of them are working away, producing the polypeptides. A polysome is the complex that is formed when we have multiple ribosomes attached to an mRNA. And again, this is to facilitate rapid translation, rapid production of polypeptides. In prokaryotes, the ribosomes can actually bind before the mRNA molecule is even complete, because there's no nucleus, so that you don't have that mRNA being produced in the nucleus, exiting, and then binding to a ribosome. It can all happen at once. So for example, we have our DNA here. You can barely see it along here. We have mRNA being made and transcribed. And then we have translation of that mRNA happening. These dark blobs are the ribosomes that are attached. Isn't that really cool? This allows for prokaryotes to rapidly respond to their changing environment, because you have to remember they're single-celled organisms, so it's very important for them to be able to respond rapidly. Okay, and lastly, recall that polypeptides must be folded in order for them to function. And we talked about that in Unit 1, where we have our, the primary is really all that's being produced here is our primary sequence, the order of amino acids. Then we have our secondary, tertiary, and maybe even a quaternary folding or structure of that protein. Modifications that fold the protein can be done by enzymes. So enzymes may remove amino acids from the ends or even from the inside, and then you're producing a completely different enzyme or a different protein. You might also get the addition of molecules such as sugars to that polypeptide, and again, giving it a different or just giving it its functionality. Many proteins are made of several polypeptides, remember hemoglobin, and the cell controls how these polypeptides are activated and brought to use. And this is a form of gene expression. Okay, so that is our translation and the different steps involved. So we have the initiation, elongation, and termination.